Hi, my name is Oliver Bell. I'm an assistant professor in biochemistry and molecular medicine at the Keck School of Medicine. My lab is interested in understanding the molecular mechanisms underlying epigenetic gene regulation. I've previously trained and obtained a PhD in biochemistry from the Friedrich Miescher Institute for Biomedical Research in Basel, and I've performed my postdoctoral training at Stanford University. I've started as an independent investigator at the Institute of Molecular Biotechnology in Austria before moving and relocating my lab to USC in 2018. We're interested in understanding the molecular mechanisms of epigenetic regulation, which involve chemical modification of chromatin structure. So what is chromatin? Basically, we know that our genes and our genomic DNA is present in our cells not as naked DNA, but in fact, it's wrapped up in a structure called chromatin, which is the combination of DNA and proteins. And chromatin provides an opportunity for regulation either by enhancing accessibility or increasing the ability of proteins to interact with the DNA, or by restricting the interaction with proteins and DNA. In particular, we're interested in understanding the mechanisms involved in epigenetic gene silencing by the polycomb repressive complexes. These form two classes of epigenetic regulators which are able to catalyze chemical modifications on the surface of chromatin, and in turn read these chemical modifications and exert their repressive activity by restricting accessibility of regulatory proteins with the DNA. Importantly, polycomb complexes are frequently dysregulated in different diseases, including cancer. Therefore, academia and also industry have identified polycomb regulatory complexes as potential therapeutic targets for therapy. And so most efforts have been directed at trying to inhibit the writer activity that is responsible for placing these chemical moieties on chromatin. And no inactivators or inhibitors of the reader activities have been developed so far. Now, in collaboration with chemists at UNC, we've developed several chemical probes which specifically target these reader activities, and we're interested in exploring their therapeutic potential. So we're looking for partners in academia and industry who are interested in exploring what is the role of polycomb repressive complexes in disease, and to evaluate whether blocking or inhibition of reader activities could be a potential therapeutic strategy or avenue. If that's something that is interesting and exciting to you, please contact us.